Hello, my name is Agnieszka Piotrowska and I'm an Alliance Manager here at Ardigen. And today with me is Kaja Milanowska-Zabel. Hello, Kaja.
Welcome.
And Kaja is our Executive Vice President and also leads all our efforts in our R&D Microbiome unit. Kaja, could you tell us a little bit more how our technology works?
Yes, of course. So the platform is designed to take the NGS data, so shotgun microbiome sequencing data, to get the information about the genomic features that are hidden within the data. So for example, we have a cohort of patients, let's take oncology patients. Mm-hmm. And we have responders and non-responders in that therapy. And we would like to know the difference that lays in the gut microbiome between responders and non-responders to get the hint about potential output from the bacteria living in the responder's gut.
Mm-hmm. So what type of information is needed for the analysis of such study?
SMS data, so shotgun metagenomic sequencing data. And that's it, from the cohort of patients, for example.
And what's the benefit of using the metagenomic information in such
Well,
case?
the benefit here is, imagine you have the 16S data. From the 16S data you can go only with the taxonomy-based approach.
Certain level.
Yeah, so certain level. And with that information you are, first of all, based on the reference that is behind that, and second of all, you will not get the deep information. With the shotgun metagenomic sequencing, you get the actual information that is hidden in the data that you receive. But important thing here is that we are not reference based. So we are utilizing the whole information from the cohort. So, for example, imagine you have a cohort of patients from a certain geographic region. You have responders and non-responders there. And in the responder's gut is a unique gene hidden that is responsible for the responding to the therapy. And if you go with the reference-based approach, you will lose that information. And with our approach, you can get that information and then you can find the genome of the bacteria that is bearing that information.
Could you explain the functional approach?
So with the signatures we are receiving, those are the genomic sequences that can be responsible for giving the certain protein enzyme. And then we are translating those information. We are translating it to get the information about the function that is behind that, to get the information about the biology, metabolic pathway, how it really works in the patient's gut.
Okay, Kaja, so how this all can be applied?
Well, first of all, you can get the biomarker for the diagnostic purposes. Mm-hmm. You can differentiate groups of people, so you can, for example, use it for early diagnosis. Mm. The other thing is you can then proceed to go to the therapeutic area. So, for example, from the one signature that gives a metabolite, you can go to a single molecule approach. Or you can go to the LBP-based approach, so produce a consortium of bacteria that can support a therapy. What is also important, from the signature that we get from the sequencing data, we also get the information about the genome of the bacteria bearing that signature. Okay. So we can also produce LBP like a monobacterial LBP or a consortium from that. So bacteria can also be used as a biomarker. So you have two different approaches with the diagnostic road and therapeutic road.
Therapeutic. Thank you. Many ways to explore. Thank you. Thank you very much, Kaja, for answering all my questions. And thank you very much for the attention, and we hope to see you soon. Thank you.
Thank you.